Boa tarde, pessoal. Para mais essa sessão aqui do IB Virtual e da nossa disciplina de seminários da pós-graduação. Hoje teremos um convidado internacional, muito importante. Eu vou deixar a professora Ana Amélia apresentar. Só queria lembrar a todos, né, que estão assistindo, se puderem subscrever o nosso canal, deixar o joinha, o like e ativar o sininho para as notificações, vocês nos ajudam a subsistir no YouTube, tá? E eu espero que vocês gostem bastante do seminário, aproveitem, façam perguntas, lembrem-se que no YouTube vocês podem colocar no chat e a gente tem uma equipe coletando as perguntas e na medida do possível, claro, porque às vezes tem muitas, né, a gente tenta endereçá-las todas ao nosso palestrante. Então, aproveitem, and I will pass to Professor Ana Amélia. She will present Dr. Joshua, to whom I thank you very much for the opportunity to stay with us today. Thank you. Ana Amélia.
Our invited professor today is graduated in Cornell University College of Arts and Science, received his medical degree for Cornell University Medical College, and completed the internship and residence at New York Hospital. Then he did clinical and research fellowships in the Division of Infectious Disease of the Department of Medicine at Einstein Montefiore, and then he joined the faculty in that division as an instructor in 1998. Nowadays, Dr. Nosanchuck is the Senior Associate Dean for Medical Education at Albert Einstein College of Medicine and the Montefiore Medical Center. He is also the professor of medicine in the infectious disease area and the microbiology and immunology. He serves on the editorial board of several journals, including the Journal of Infectious Diseases, Infection of Immunity, and Several Frontiers, and so on. He has published over 300 scientific papers and several book chapters. He is an elected fellow of the American Society of Clinical Investigation, American Academy of Microbiology, American College of Physicians, and Infectious Diseases Society of America. During the last years, he has received several awards associated with his outstanding performance in the medical field. But the best part of his resume is his cooperation with Brazilian research, both in America and in Brazil, as Rosana Puccia, Marta Rodrigues, Célia Almeida, Carlos Taborda, Leonardo Nimrich, and me, of course. So, Josh, thank you to accept our invitation to this lecture. It's a pleasure to receive you in our university. Now, the talk is with you.
So thank you so much for that incredibly kind introduction, Ana Amélia. I'm very grateful to have the opportunity to speak with everybody today. I have to disclose that some of this work is funded by the National Institutes of Health, but I do also want to echo what Ana Amélia said about Brazil. So the first person that I worked with from Brazil was Carlos Taborda, and he was a postdoc at the same time as I was in Arturo Casadoval's lab. And I also got to interact with Professor Travassos, who unfortunately passed away this year. I also began working with Rosalie Zancopa-Olivier in Rio. And through her, I started having some students in the lab. One of them is Alan Guimarães and his now wife, Mariana, who met in my laboratory. I had the opportunity to work with many other really wonderful folks in mycology in Brazil. But Carlos introduced me to Marcio Rodrigues, and he and I have had several procedures together. And he introduced me to this gentleman, Leonardo Niemrichter, who has a student who completed a postdoc in his laboratory and is now my postdoc, Danielle Zamit. The other person who Marcio more recently introduced me to is Liz, and she's in Curitiba, where Marcio moved to. And I'm showing you some of these people because they're pertinent to the work that I'm presenting today. But what about Brasilia? Well, I have been to Brasilia. This is kind of out of focus, but you can see that I was here with several of people you may know. And I'm very proud of having published with several of the faculty at your institution. So I do want to say obrigado because Brazil has been incredibly important in my career. So now we're going to turn to the talk, and that's on Candida auris. And what about Candida auris? Candida auris is an infection that you may not know as much about, because fortunately Brazil has not had much of an encounter with this pathogen. It's very new. It was only described in 2009. And why is it so important? Well, it's important because it has remarkable drug resistance. In fact, a large number of isolates are multi-drug resistant. It is very resilient in the environment, meaning that it can live in water, it can live on surfaces, and it can live inside of us. The mortality rates on average are about 50%, which is very high for an infectious disease. Because it is so tenacious in the environment, it causes outbreaks in healthcare settings where it gets spread from patient to patient. And the other thing is that regular identification by standard microbiology techniques is very difficult. So we do it at my institution by MALDI-TOF, and it's easy to do, but not everybody has MALDI-TOF or sequencing readily available. So this organism was a surprise. We didn't expect this. And the other thing is, it's really scary as an infection. We've seen it on all continents except for Antarctica. And you can see here that Brazil is still largely untouched by this infectious disease. Unfortunately, New York in the United States has had the most cases in my country. So we have almost twice as many as in Illinois. And this data from the CDC hasn't been updated since July because they've been busy tracking SARS-CoV-2, but we know that we now have over 600 cases in New York State. So again, the global mortality rate is about half of the people that acquire this infection do not survive. And that's partly because the people who have this disease are very sick. They're in the intensive care units. They have immunosuppression. They're getting different kinds of surgeries, and they're on ventilators. It's also hard to get rid of. It's like a spore in terms of how tough it is in the environment. This is the first fungus to be designated as a global threat by the CDC, to underscore how important this is and how dangerous it is in the healthcare settings. In the United States, most of our isolates are resistant to things like fluconazole, and 33% are resistant to more than one type of drug. This really varies depending on where you are. In Southeast Asia, almost 100% are resistant to fluconazole, and they have a 40% resistance to amphotericin B. It's not very common for fungi to be resistant to amphotericin. And in some parts of South America, they have a high rate of echinocandin resistance, which is very scary because that's the primary drug that we're often using to treat these infections. It is so hard to get rid of. It's hard to get rid of in the environment, but it's hard to get rid of on us. Once it gets onto a patient, you can see here, only 20 out of 300 of the patients we had in New York were able to clear the organism, which means that they can spread it to other patients, and they can also have the disease more than once. They can relapse. So very, very scary. Because it happened in New York and we saw so much of this, we started working on this fungus fairly early, and we started working on it in about 2017. And this is the first paper that my group published on this. And I want to show you that this gentleman over here, Daniel Zamith, who came from Leonardo's lab, were the people that were involved in this work. But also 2 other individuals, Ernesto Nakayusu, and Ernesto did his PhD in São Paulo with Igor Almeida. He is a Brazilian scientist who's now in Washington State. So we had 2 organism isolates that we worked on in this paper, and they were from my hospital. One of them was extraordinarily resistant to fluconazole. We could give 1,000 micrograms of fluconazole, such that the media was almost so turbid it was almost no longer liquid, and the organism still grew. It was also resistant to echinocandins, to caspofungin. And the other one was very susceptible. And we also compared this to a standard strain of Candida albicans that we've worked with in my laboratory. When we did transcriptomics and other analyses of the whole yeast cells, we found dynamic differences. You can see here very clearly that where you have a reduction in signaling, you had a marked increase in Candida auris and vice versa. So very, very different biologies. And just to take a snapshot of some of the things that we saw that gluconeogenesis was very differentially regulated in Candida auris compared to Candida albicans. Candida albicans had much more abundant activity in gluconeogenesis, ribosome activations, and other areas, whereas Candida auris, the TCA cycle, and especially lipid metabolism, was highly upregulated. So even though they're both Candida species, it's like saying a gram-positive and a gram-negative organism, very different biology. And the resilience that you start to see here in terms of some of the functions of Candida auris in some ways explain why it is so metabolically facile in the different environments where it's facing. Candida albicans really only lives in humans, but Candida auris, as I said, can live in the environment and it can live in us. So ergosterol biosynthesis, extraordinarily important. Again, ergosterol is the target of both the azoles and the pollines. And so the fact that two of our major antibiotics are targeted in different ways and changed in its regulation in these species is very important. And when we looked at 11 of the 19 ergosterol synthesis enzymes, we saw that Candida auris had much more abundant of ERG24, ERG11, ERG3, ERG2. All of these were upregulated in Candida auris compared to Candida albicans. So very, very different biology between the two types of yeast. Lipid biosynthesis, very different. You can see here that the triglycerides, for example, are significantly upregulated in Candida albicans. But we had really these lysophospholipids and sphingosine bases that were significantly upregulated in Candida auris. So what did we see here? We use this multi-omics approach to begin to differentiate what's happening in these two species. And we really found that the resistance processes were multifactorial. So they were different because of the metabolism, they were different because of cell wall formation and things like efflux pumps. And you can see here that in this one here, the CDR1, that our resistant strain, the MMC1, was much, much more activated compared to the sensitive strain or to Candida albicans. So the way these organisms respond is very variable and extraordinarily different from Candida albicans. So some of you know that we've been very interested in extracellular vesicles, and this work really began in the field with fungi with Marcio Rodriguez during a visit that he had to Albert Einstein back in about 2006, 2007. And since that time, our laboratory and others have shown that diverse fungi, Ascomycetes, Basidiomycetes species, produce these extracellular vesicles. These vesicles are typically about 50 to 1,000 nanometers in diameter, and they are basically virulence bags. They have all sorts of things inside of them, many of which have been linked to virulence. So things like melanin inside of here. In Cryptococcus neoformans, the glycopolysaccharide is packaged within these vesicles. And people like Rosanna Puccia and Liz Alves have shown RNA is inside of these vesicles. These vesicles are really interesting because they do so many different things. These vesicles can change how macrophages are activated, how activated they are, and they can enhance their sidality against fungi. They can change polarization of macrophages. The dynamic alteration in vesicle production is incredible. Just having antibodies binding to the yeast cell surface can shift what is being loaded into these vesicles. So it is a very highly regulated process that we don't fully understand yet, but we know that it's dramatically regulated. The other thing is these vesicles can change outcomes between the host and the pathogen on a larger scale. So they can protect against Candida albicans infection. These vesicles also could be used as a vaccine platform, and that work was just published by Leonardo Nimrichter's group very recently. So when we were thinking about Candida auris, we figured that the extracellular vesicles that were being released were going to, in some ways, regulate the biology of the host pathogen interaction. So we assumed that Candida auris, like other fungi, would make vesicles. And we assumed that they would be different from those generated by Candida albicans. And then we wanted to see whether they, in fact, would regulate the host pathogen interactions. So this work, again, was done with Leo and Ernesto, but it was also done with Marcio Rodriguez and with Radamés Cordero, who is a US scientist, but he spent a couple of years down in Rio de Janeiro with Marcio and Leo through one of the Brazilian Visiting Scientist Awards. We also worked with Liz Alves on this work. So the first thing we found was that, yes, in fact, vesicles are produced by Candida auris. You can see here the lipid bilayer, and there's some differential density within these vesicles. Interestingly, we found that MMC1 had very different sized vesicles, both in the smaller population and the larger population, compared to both Candida albicans and MMC1. We saw that there was differential amounts of protein and sterol, and that they were different between the two species as well as between the two isolates of Candida auris. What we also similarly found, as we saw in the cells, were that things were extraordinarily different in terms of the proteomics. So one of the biggest things that we noticed was that these phospholipases and other lipid biology markers are differentially regulated and packaged into the vesicles of Candida auris compared to Candida albicans. So things like these lipophospholases were significantly upregulated, where just a regular phospholipase was highly regulated upward in Candida albicans. And this is, again, another way of looking at this, where we really saw very significant dynamics between the whole cells and the vesicles. And the fact that what was significantly upregulated in the whole cells is different from what we saw in the vesicles shows that this is a regulated process, that it's not, again, just garbage in, garbage out, that this is a dynamic and there's a purpose to the generation of these vesicles. When we looked at lipids, you also see, again, very marked differences between the Candida auris strains and the Candida albicans strains. And in particular, we found this interesting ceramide in the resistant strain. And when you have these differential ceramides, it goes towards very distinct differences within the cell wall. So cell wall, these contents in the vesicles, are they being delivered to the cell wall, or are they biologic compounds that are being released out into the extracellular milieu where they could be biologically active? And as I said, extracellular vesicles have been shown to also carry RNA, and so we looked at that. And there was significant numbers of RNA, particularly with biosynthetic processes and stress responses. And you can see here that there was significant difference between what we saw in the vesicles in blue versus the red in the cells. So what was significantly upregulated in the cells was not upregulated in the vesicles, and vice versa. And we also figured out that the majority of the RNA were tRNAs. And this gets to some of the differential ontology groups within the vesicles compared to the cell. So in the cell, we saw carbon metabolism and glycolysis, whereas up here, we saw much more stress response. what about the biology? What does this mean? Does this translate to anything important? So one thing we did was look to see how the yeast would change in terms of their adhesion to epithelium if vesicles were present. And what we see here is that for our more resistant strain, when endothelial cell layers were challenged with vesicles before the yeast arrived, we had significant increases in the amount of the Candida that labeled there. And you can see here, again, the Candida is in red, how many more yeast cells are attached to this cell layer compared to the absence of the extracellular vesicles. However, we did not see differences in phagocytosis and killing. And that may be just because of the cell type that we were looking at, or maybe it's just the biology of the disease. So we need to look at that a little further. We found that these extracellular vesicles stimulated dendritic cells, which we knew from Candida albicans literature occurred. And you can see here that both the susceptible and the resistant MMC1 significantly activated dendritic cells. So whether it's from Candida albicans, whether it's from a susceptible or a resistant strain, we saw that these extracellular vesicles activated dendritic cells. So in terms of this portion of the presentation, we proved that Candida auris released extracellular vesicles. We showed that the size of these vesicles, as well as the contents, were significantly different between Candida auris and Candida albicans, but there were also differences between the different strains. The data that we have shows that vesicle biogenesis is highly regulated. There are biological effects of these vesicles. They change adhesion and they can activate bone marrow-derived macrophages and dendritic cells. So one of the things that we thought of is what else is happening here in these infections in humans? So we know about activation of macrophages. There's information from Janelle Nett's laboratory in Madison, Wisconsin on interactions with neutrophils where the organism activates the extracellular nets that take place. But what about T cells? T cells are very important in regulating Candida infections with other species. And there really was very little work on T cell biology and Candida auris. So this work, again, was done in my laboratory. This is Danielle, who, again, did most of this work. And we found, very interestingly, that these vesicles, as we've shown in other experiments from Candida albicans, activate T cells, activate other cells. So here is the first time we're really looking at T cells. But look what happens with either direct or indirect activation with either MMC1 or MMC2. We see a marked reduction in activation. And in fact, when we were doing our flow, you can see here, if you activate a T cell with LPS, you have this change in the fluorescent intensity. Very similarly, we had beautiful activation with Candida albicans extracellular vesicles, but with our auris vesicles, we saw almost no activation of the T cells. We did freeze thaw and we thought, okay, what types of products are involved with this? And after freeze thaw and just using free proteins and whatever else was in that thaw cycle mix, we saw that, again, Candida auris, particularly the MMC1, had no activation. So then we did different things. We did proteinase K, RNase, DNase, and these led to no changes in activation. So there's something in these that is regulating the suppression of this interaction. And what we're doing right now is we're looking at whether lipids, like that ceramide I showed you before in the extracellular vesicles, could be involved in the biology of inhibition. So now we're going to turn to drug resistance. And we talked a little bit about amphotericin and azoles and even the echinocandins. And the echinocandins are the primary drug that's recommended for treating these infections. But can we take these organisms that I showed were incredibly metabolically malleable and evolve them either with fluconazole or a pauline or itraconazole, posaconazole, or echinocandins? So we set up this process with this gentleman here who's Attila Gaxer, who's another scientist who did his postdoc in my laboratory, then went back to his home country of Hungary, but is collaborating actively with several individuals in Brazil. So I think he's given talks in your group as well. So this is work that Attila and I are looking at. And I do want to acknowledge that this is not totally novel because we used a similar process to do microevolution studies with antibiotic stress in Candida parasilosis, another organism that we work on. So what we found here was fascinating, because if we had a strain that we evolved in a specific antifungal, there were marked changes that happened to others as well. So you can see here that the wild type of Candida auris, of this particular one, to fluconazole was 12. And if we evolved it in fluconazole, we found that there was an increase. But we also saw increases in other types of situations. So we would see that, for example, micafungin went from 0.125 to 0.5. So this is not quite resistant yet, but it certainly changed. Another azole, however, gets significantly upregulated just by treating with the fluconazole. What's really interesting here, though, is that fluconazole evolution led to high-level resistance of amphotericin. So we were evolving it in the setting of one antifungal and generating resistance to other antifungals. Very, very concerning. What we also saw was that we had some interesting changes in sterile composition. In the setting of posaconazole evolved strains in particular, where we had these increases in certain types of sterols, which in general are felt to be somewhat toxic to cells. Yet you have high levels of these new sterols, and the fungus itself in the evolved situation is growing perfectly fine. So again, it gets to the metabolic plasticity of Candida auris, able to survive very diverse stresses and adapt very quickly. Does this evolution translate to more than drug resistance? Usually when you have an organism that generates resistance, particularly, for example, if you take HIV as an exemplar, HIV, when it has mutations, the fitness of that virus decreases. What we saw here is that with certain of our strains, and in this case, it was with the fluconazole evolved and the voriconazole evolved in a Galeria model, we saw that these evolved strains had increased virulence. So not only did they develop drug resistance, somehow they gained virulence in this model. We also looked at this in mice. And in fact, in mice, we saw similar types of findings. And in this case, we're also seeing posaconazole leading to increased virulence. So here we have the kidney, the liver, and the brain with these organisms having much higher CFU. So what we're doing currently is we're evolving these in the setting of different antifungals. So we're looking at the echinocandins, we're looking at different azoles. And again, we have most of the azole strains in hand. And we're also looking at the pollines. And we're trying to see what leads to cross resistance. We very strongly feel that this is involved with sterols and lipids, which is the area that we're really focusing on. And then we're, again, trying to see what the biologic implications of this are in terms of host pathogen interactions. However, as is clear there, I think, we still don't know very much about the effects of what these antifungals are doing on this fungus. As we mentioned, caspofungin is the standard. And so we took this and said, what does an echinocandin do in terms of the transcriptome, both in the yeast cell as well as in the extracellular vesicles? And this work was done with Liz and with Marcio, but not with Leonardo. Actually, we would love to include him, but he's not included in this particular work. And then my friend Ernesto, the gentleman who did his PhD in Sao Paulo, and my postdoc Danielle did this work. So we figured that this antifungal would silence some pathways in order to respond to the stress. And again, we posited that based on the really rapid adaptations that we've seen in our other models. And we figured also that the extracellular vesicles would have differential loading in response to these changes. And primarily that we would expect to see signaling stress and things that were pertaining to cell wall maintenance. So what we first saw was that there was a slight change in the size of the vesicles, but it was not significant. So you can see here that there was a slight change, and that there were slightly bigger vesicles in both of the two strains that we looked at, one of them from the CDC and one of them from my institution. So you see that there was a shift as well in terms of the amount that was being produced and the size in the setting of caspofungin, so response to stress. So this is all very new data, so I don't fully understand it yet because we're still talking about it. I actually had a call earlier today with Liz to try and figure out some of this information. But what's absolutely clear here is that when you look at the changes that take place in the cell in response to this antifungal, that you have very distinct settings for where regulation is taking place, as the two strains that were treated with the antifungal really track together compared to where they are in the absence of the antifungal. And what you see here in terms of enrichment are things like sugar metabolism, cell cycle. And just to put in here that these were very large reads that we were able to generate that allowed us to do some very nice mapping that we're still working on. We found that there were really highly distinct types of RNAs that were packaged in here. And similar to what has been shown in other species, we saw lots of different types of RNAs. And again, we saw that you had changes in ribosome and cell wall in the vesicles. So we wanted to see what things were being packaged in the yeast versus in the cells, similarly what we were looking at in our other studies. And consistent with those findings, what was highly upregulated and enriched in the cells were not as enriched in the vesicles, and what was highly packaged into the vesicles was not as highly packaged into the cells. This is, again, really underscoring that the generation of extracellular vesicles is a highly regulated process and one that we really need to understand better. This just shows you some of the differential distribution of the non-coding RNAs, and that they are, again, distinctly different in the extracellular vesicles compared to in the cells, whether or not there was treatment with the antifungal. So very, very different biologies. Yet they're similar in terms of their responses to the different strains that we were looking at. So what we saw here is that these genes, particularly that were stress response, cell wall, cell regulation, plasma membrane, were significantly upregulated. Metabolism genes, interestingly, were downregulated. We saw that these vesicles were highly associated with stress response, which is what we anticipated, and that the vesicles are participating in the response to cell wall maintenance and stress signaling. What's also really important is that these really descriptive works give us a lot of information for other targets that we can look at. So can we look at lipid metabolism? Is that an area that makes a lot of sense? And based on what I showed you with the evolution, as well as some of the findings here, it suggests that's a rich area to explore. There has been CRISPR-Cas9 applied to Candida auris, We've had some difficulty achieving that. There are not many mutants that have been described. There's also targeted gene disruption, but that too has been very challenging in Candida auris. So once we get a better system and get more experience with gene disruption, we should really be able to dissect out what the meanings of some of these regulatory changes are. I'm going to take a few minutes to talk about a different fungus. And I just am looking at the time, and I guess I've gone very quickly. And I think that's in part because I'm not seeing anybody and no one is asking questions yet. But I do want to take time to talk about some work in histoplasma because it does complement some of the other things that I've shown you so far. So histoplasma, this work was, again, mostly done with Ernesto and Daniel. And Alain Guimaras and Leonardo Nimrichter and Marcio have worked on this as well. So histoplasma in the United States is very important, particularly in this part of the United States. But you can see here that Brazil has areas where histoplasma is also very, very important. In the United States, we have about 500 million infections each year. But what's interesting is that does not correlate with high numbers of hospitalizations. And we have about 350, 400 deaths a year. I would put that into context, though, because in the United States, we have under 500 deaths a year from tuberculosis. So the fact that this is causing almost 400 deaths a year is not that different than what we see with tuberculosis. So in terms of mortality, it's an incredibly important infection in the United States. So I've talked a little bit about lipids in this presentation, and lipids are really truly fundamental to biology. Maintenance of the cell membrane is essential for homeostasis, but I also told you that they're targets of our main antifungal drugs in terms of the azoles and the amphotericin Pauline types. So really important both for the host as well as for us as clinicians. Understanding what's happening with lipid metabolism is highly essential for developing new drugs and targets and really understanding the biology of the disease process. So we've been studying lipids in histoplasma for a while, and we recently built this comprehensive map to look at metabolic pathways, where we tried to really take a lot of various omic data and put them together into a metabolic map. And we did this because we wanted to understand what was different in histoplasma compared to other fungi, but also to see if there were targets that were important in pathogenesis, as well as new areas to study for drug development. So this is a really quick way that we did this. We used something called MPLEX. And so by doing this, we were able to do lipidomics and proteomics and generate huge amounts of data from our samples. I'm going to jump to the map, and you can see how this map is fairly intricate and very rich in information. And if you look at different parts of it, you can find different things. So for example, what we found in histoplasma is that the type of ceramides that were being generated are these hexosyl ceramides, whereas in Saccharomyces and most other yeast, you have a very distinct difference in the ceramides that are being formed. So this is an area of distinct biology and one where developing antifungals might be an interesting way to pursue. The other thing is we found an odd platelet activating factor. And so platelet activating factors are molecules that lead to the activation of platelets, and you get platelet clumping. It's something that happens in sepsis. It's something that in a local as well as systemic level in a disease can cause significant host changes. And so we found this molecule, and then we decided, okay, we've proven that by mass spec and other techniques that this is consistent with a platelet activating factor, but does it have any biological activity? And in fact, we showed that it does. So if you have your platelet activating standard and you have our fractions where we found this by mass spec, when you apply different concentrations of these fractions, you see platelet activation taking place. Whether you're looking at it through TNF alpha production or IL-10 production. And if you have an inhibitor of PAF, you have the abrogation of that response. And you can see again here with our PAF and our fractions in terms of activation. So it is biologically active. So we built this incredibly detailed, comprehensive map of the metabolic pathway. And this allowed us to see distinct differences in biosynthesis, which gives us clues for drug development. We also found some interesting biology in terms of that platelet activating factor, which could potently regulate responses to this infectious disease. So really powerful information that you can leverage. So I do want to acknowledge the people that I presented this work with. So in particular, Daniel from my laboratory, Leonardo Niemrichter, Marcio and Liz, Alan, Arturo Casaval, who has been my mentor for a very long time, and Radamez, who's still with Arturo again, and then Ernesto in the Washington State, and Attila in Hungary. And I didn't present any of the work that Anna-Melia and I have been doing together, but Anna-Melia is incredibly special for me, and I really appreciate the interactions that we've had. I've learned a lot from her. I've enjoyed visiting her, whether it's in New York or in Brasilia. So thank you very much. And we have plenty of time for questions because I did go fairly quickly, even though I thought I was going to have not enough time. So thank you for your attention.
So thank you, Josh. I think that you can take off your, yes. So thank you for our explanation. Halloween is only on Saturday, but you did a scare presentation about the Candida auris. Oh my goodness.
You make your hair stand straight up.
Yeah. Yeah. It's terrible to think that we are living with this pathogen that we can't treat. And worst, when we treat, you can increase their resistance to the drugs.
And the virulence too.
Yes. So it's terrible.
In times of COVID-19, it's nice to see that we got much more of them.
Yes, it's not the worst scenario. Yes. So I will start with a philosophical question, because Arturo was invited to Susanna to talking about the fungi and the warming, the global warming. And they mentioned that the Candida auris were coming from this increase of the temperature. So considering this, what do you expect from the pathogens that you are living, and what you can think about the pathogens that are living with us with the increase of the temperature, or what you can come to us?
So I think that's a really important question. And first, I think we have to think about what these organisms are doing today. What are they seeing in nature? And one of the activities that I was involved with very early in my postdoc in Arturo's lab was looking at the interactions of fungi with environmental amoeba. So we found that with cryptococcosis, and these amoeba, the Cryptococcus could win. The Cryptococcus could use these environmental organisms as food. So that was true for Histoplasma and Blastomyces. But what's really important also is that amoeba are basically very similar to macrophage. So these organisms are encountering very similar situations in the environment to what our host response throws at them. So whether it's hemolymphogalaria or whether it's the inside of an amoeba, and people like Alan and Niterói are still working on a lot of this amoeba interactions. It's a boot camp for these organisms in the environment. So they don't need that much more of a change for when they encounter us to be able to survive, especially if they can survive at our temperature. So Candida auris is a great example of something that may very well have evolved with the increases in temperature. One of the things that I showed in my talk is how metabolically resilient this organism is. And because of that, it's able to adapt in many different ways. And it's able to grow at 40, 41, 42 degrees. So it's something that really has a lot of the changes that were necessary to make that jump to us. The other thing is that we're seeing a lot of other new infectious fungi. So the blastomyces, there are now about 5 or 7 different species of blastomyces. There's blastomyces helicus in places in the United States that we never saw blastomyces before. There are blastomyces species in Africa that we never knew about. The emergomycoses in the southern part of Africa are now the most common dimorphic fungus in patients with advanced HIV infection. You know, we didn't have, and it's called emergomycoses because it emerged out of nowhere. It was totally surprising. I do think we're going to start seeing more different fungi than we've seen before. These fungi are very clever. They have massive repertoires of genes that they can use to adapt to different situations. I've always been astonished that we don't have more fungal infections than we have. And the other challenge with fungal infections is that we don't have many drugs to treat them with. We have some very interesting new ones that are coming for the first time in 20 plus years. But for the most part, you know, you have very few choices for some of these infections.
So Josh, we got some questions about Candida auris biology. People are asking if they form biofilms, for example, and if they are haploid or diploid, and if the techniques that we already have that function to other fungi will function to these fungi. What do you think?
So biofilm is a great question, and it does form very intense biofilms. It's not as thick as Candida albicans biofilms, but Candida parasilosis biofilms are also not as thick, and they too are incredibly tenacious and highly associated with the pathogenesis of the disease. We also know that in biofilms, the MICs, the ability of regular antifungals to kill is much reduced. So very important. The other thing that's very distinct about Candida albicans is that it forms multiple types of cell morphologies. So it is a yeast, but in tissue, it transforms into a hyphal organism. So it can form long pseudohyphae, it can form yeast, it can form various morphologies. And that's a challenge for the immune system to face different types of forms. But with Candida auris, it does not form pseudohyphae except in very rare instances. So in a I can't remember exactly what the very high stress media, it formed from some very short pseudohyphae. And then there was a mutant, I think it might have been to the HSP90. One of them formed some short pseudohyphae also, but not true pseudohyphae and certainly not hyphae. So,
and how about the genome? Is that haploid or diploid?
So it is diploid. So it makes it more complicated again to do the disruptions. And so, you know, glabrata is much easier to work on than auris and parasilosis. And so it's been more challenging because of that.
Let me talking about the ergosterol, because you told that there are difference between Candida albicans and the auris, and then in the vesicles for the resistance is you have a higher content of ergosterol. Okay, but what's the size of the vesicle? Because in Larissa that is watching us showed that for Cryptococcus neoformans, when we block the ergosterol synthesis, the vesicles are higher than the mutant. And in the Candida auris, the vesicle is lower than the Candida albicans, or no?
So it's very interesting because it really depends on the isolate that you're using. Some of the isolates have very similar vesicle size. So the smaller population around 50, and the larger one around 150, 200. The MMC2 one that I showed you, the susceptible one, was almost identical to the Candida albicans. The MMC1, the one that's more resistant, that one, the smaller ones were more like 100 nanometers in diameter and more like 300 or 400 nanometers for the larger size. So there were differences that we saw. The interesting thing is, depending how you grow them, depending on the media, you can change sizes, as you know from your work with Cryptococcus and our work with Histoplasma. How you grow them can really change the size of these vesicles. So there's always a question between the biological significance of what you have in your laboratory versus what's happening in a patient. But the fact that there are differences suggests that there will be differences in other conditions too.
Okay. We have another questions from the public here. Let me see. They are asking about how do you proceed with patients admitted to the ICU diagnosed with SARS-CoV-2? Is there any prophylaxis to prevent candidemia or other fungemias?
So one of the things that's been very interesting about COVID-19 is antimicrobial stewardship. So in our hospital, at the very beginning, we saw that everyone was getting the standard for community-acquired pneumonia, which here is ceftriaxone, a beta-lactam, and azithromycin, which is a macrolide. And we very quickly said there's no data for this. They have a pure viral infection. People were worried because of influenza. A lot of the early work with influenza was that people were coming in and they would have pneumococcal pneumonia, or more recently in the US, we have a lot of Staph aureus infection. That typically doesn't happen at the same moment. It usually is days or weeks later that you'll have this secondary infection. So even acute influenza should not necessarily need antimicrobials. So in our hospital, we very quickly put some restrictions on use of antibiotics in these patients. And what we subsequently found in our studies is that only about, I think it was 5 to 7% of our patients had a secondary bacterial infection. So very low numbers. And we were able to tell what those individuals are because, instead of just having the appearance of a viral pneumonia, they would also have a very dense pneumonia on top of it that's localized to a single lobe, or other findings that were very much consistent with a bacterial process. So at our institution, we really shut down the use of antibiotics. We just had our big ID week, our International Infectious Disease meeting. And one of the presentations that I moderated was from the Veterans Administration. And they had over 42% of their patients getting antimicrobial therapy, yet they also had a very low real infection. So they didn't do a good job in that setting of restricting antimicrobials. We have seen fungal infections. We've seen Candida albicans, we've seen Candida parasilosis, Candida glabrata, and we've even had a couple Aspergillus fumigatus. I think both of them were fumigatus. Those individuals were in our ICU. They were the same type of patient where if they had other significant complications, multiple central lines, on the ventilator for weeks, surgical procedures, so they were high risk for an infection regardless of the COVID-19. So we have not been using prophylaxis. We've just been very carefully monitoring the patients. That being said, we also have been less intense in terms of checking on patients. So when we had the height, so in New York City, the Bronx was one of the highest hit areas with COVID-19. We had more disease per resident than any other part of New York City. My hospital was at almost 200% capacity at one point. We had, unfortunately, several thousand deaths in my hospital system from this. We had well, now it's much more, but we had over 12,000 survivors, so that was even better.
That's good.
But the mortality rate of those who we admitted was 15 to 20%. So very significant mortality. We for infection prevention reasons, instead of having your team of like 5 people go and see the patient, you would have one person go and see the patient. And that was to reduce the interactions that you had with the virus. And it was also to reduce fatigue of our faculty, because the nurses, the doctors, the medical residents, the nutrition, they were all working so hard that trying to make them work a little less hard was very helpful. So I do think that some things probably were missed because you had fewer people looking. But what I would hope is that those few people that were looking a little harder. So
and
you know,
better, because it was not so tired.
So the other thing that is really important is that we were monitoring inflammation much more than we usually do. So most of the time when someone comes in, we just look at their white blood cell count. We maybe get what's called a C-reactive protein when they first come in, or a procalcitonin, maybe, depending on what it is. And with these patients, we were monitoring procalcitonins, we were monitoring their IL-6 levels, we were monitoring all sorts of parameters. And so we knew if somebody was doing something, that it was getting better or worse. And so we were able to more rapidly change management on these patients because we were so closely following the immunology of these individuals, which is a new way of looking at things. It's much more expensive, but we were able to be much more proactive in our activities. And again, our mortality rates early on versus what they are currently have massively changed, because we now know when to give steroids. Steroids are the only medication so far that has absolutely been proven to change mortality. So we know when to give it. We don't give it too early, because if you don't need it, you don't need it. And steroids have very bad activities too. So we know when we need to give it. Anticoagulation, we saw a lot of people die of clotting diseases, either pulmonary embolisms or strokes or other really bad things that were happening. So very early on, even in mild disease, we often will give even an aspirin to somebody with this disease. So we have many different interventions compared to before. But in terms of antifungal prophylaxis, we typically will not do that. We start thinking about it once a patient's been in the ICU for a while, though. We certainly do.
So I think we have 2 questions about the extra EV. Can you do, Ana Amélia?
Yes, one of them is a question for a Brazilian research. I don't know if you know, it's Marcio Rodrigues. And he asked for you, do you think that Candida auris produce EVs in vivo?
I absolutely think it does. One thing that was very interesting today, and some very fresh data that Liz, Marcio's colleague, showed me, was some scanning electron microscopy images of treated and untreated Candida auris with caspofungin. In the setting of caspofungin, where there was a stress response, she sees what looks like vesicles on the top of the cell. So I think if you really stress these, you're going to see even more vesicle production. And that's what we saw in the experiments that we were doing. When we were stressing Candida auris with caspofungin, and even just the quantitation of the vesicles, we had a lot more vesicles. So I do think we're going to see it in disease because of what we're seeing in our artificial experiments right now. How could we prove it? Well, probably one of the easiest ways to do it would be to do a catheter type of model, which we've done with Candida albicans. But when we did that, we weren't looking for vesicles. But if you colonize a catheter and put it into a mouse or into a rat and leave it in there and have blood flow and things like that, and then you take out the catheter and then you section it and do EM, if we could see the same thing there as what Liz saw in her Petri dish over the last couple of days, that would go a long way. But thank you for that question, Dr. Rodriguez.
There is another question.
Yes, about the EVs. Do you think that these EVs are good for the fungi or for the host?
So I think it depends on the organism, and we're still trying to tease that out. what I am most excited about with the Candida auris EVs and the host response is what I was showing with the T cells. And if this is really suppressing the cellular immune response, it might be explaining why we can't get rid of this organism from the body. So if you can't mount an appropriate T cell response, we know in Candida albicans that these individuals have more significant disease and it's harder to get rid of the fungus. So if this organism by itself is suppressing T cell activation, that might be part of the biology that allows it to survive and colonize and cause disease if it gets the right opportunity. So I do think in particular with auris that vesicles are part and parcel of the pathogenesis of disease.
Yes, and the content of that change, is that right? The content of the EVs change along the disease or the medium that you use in vitro. Do you think that it's possible?
Again, we haven't looked at Candida auris in terms of those stresses. We've looked at Histoplasma most extensively, and your partner Amelia has really looked at it in Cryptococcus with 10% SAB versus regular SAB, and showing very different biology in terms of host pathogen interaction. So I think that these are really important areas to look at. They're understudied and they're difficult. They're challenging studies to do. But we're hoping to continue along this way and get more answers in the future.
And I will make a question by me. Do you have any diagnosis already set for this pathogen?
So for Candida auris, there is no biomarker that we have. Basically, when you get it in a culture, then you can do basic biochemicals. The problem there is that you often misidentify it as Candida hemoloni. So you really need to do either sequencing of specific gene regions or MALDI-TOF, which is very good, but not everybody can have MALDI-TOF in their institution. So, I know in Brazil, many of your institutions now do have this as part of their clinical labs, and the central governmental lab, which sends everything to their central labs, does it. But again, if you're sending something and it's got to go by mail, then it takes 2 days to get there. And so diagnosis can be very delayed. The good thing in some ways is that, again, the echinocandins are first line for candidemia. So if you know that there is a yeast, most of the time it's going to be Candida. And so using an echinocandin is good. We do know that there, again, in South America, if the people from Colombia, Argentina sneak over your border and they have Candida auris, they may have 10% or more resistance to echinocandin. The other thing that's really important about echinocandins and yeast is that if you just have a diagnosis of yeast, it could also be Histoplasma. It could also be Cryptococcus. And guess what? Echinocandins don't work for those infections. So it does get complicated. And I've seen many times when it's been 1, 2, 3 days of somebody on an echinocandin when it has been Cryptococcus or Histoplasma. So it's really important to work with our clinical laboratories and think about what the underlying immunology is of that patient. So if it's a patient who has a renal transplant and they're on a few different immunosuppressants and the person's having some fevers and they grow a yeast, but it's on day 3 or 5 of the hospital stay, it probably isn't Candida auris or Candida albicans because those yeasts grow fast. But Cryptococcus grows much more slowly. So you need to talk to your clinical colleagues. You need to think about the disease. It's a multi-step process, but it's a very interesting process.
Yes, but it will be nice because of the mortality of this fungi. So do you have any other question, Amelia, from yours?
No, I think that it's enough. I think
I know, but
a lot of things to think about your life. And to stay at home because you can find
never go out again.
Yeah, so thank you very much, Josh, to spend this time with us and this really nice talk. There are a lot of people saying that is a really nice
Yes.
talk, and
A lot of
thank you.
congratulations.
Well,
A lot of
I miss my Brazilian friends very, very much.
I think everybody was there at the YouTube. They just say, hi, Josh. Hi, Josh. Nice talk. Nice talk.
Hopefully they're not making fun of my COVID hair. It's very big.
Yes, yes.
Okay. So
Thank you so much.
Thank you so much. We are very thankful for you. Okay, thank you for your
Okay.
lecture.
Obrigado.
Yes. De nada.
Okay, bye.
Bye, everybody.